Here at ESC 2015 in a hotline session, we have the first of its kind data from 2 independent studies that have identified a gene associated with life-threatening abnormal heart rhythms that can lead to sudden cardiac death. And we're here to talk first about discovery, and I'm with Dr. Heiner Wieneke, who is a professor at the University of Essen. So the first study presented used ICDs, correct?
Yes.
To monitor. So tell me about the whole concept here first before we get into the data.
The first study, we looked in patients with ICD who are at risk, patients with defibrillator who are at risk of sudden cardiac death. We looked if we could identify a genetic marker for the occurrence of ventricular tachyarrhythmias. And we included 1,100 patients, and the defibrillator is somewhat like a continuous holter over 2 years. And so we could monitor the patients for 2 years and look if ventricular tachyarrhythmias occur, and then we could relate our genetics marker to the occurrence of ventricular tachyarrhythmias. And this was the discovery study. We find 2 genes encoding for G protein subunits that are proteins involved in signal transduction, and these 2 SNPs were related to the occurrence of ventricular tachyarrhythmias, and the patients who were homozygous for the variant gene had a 50% increased risk of ventricular tachyarrhythmias in this first part of the study.
Wow. So now let's go to the second study.
And then in the second study, that was the Oregon SUD study, it's an American study. The lead investigator is Samit Chuk from University of California. And this is an observational study collecting all cases of sudden cardiac death in the Oregon area. They had about 900 cases, and we looked if our SNPs we identified in the first part were also predictive for sudden cardiac death in his study. And we saw that one of the both markers shown in the first study, significant, was also significant in the Oregon SUD study.
So in Oregon SUDS, you have blood samples from 1,335 patients. So that's a good group of people. You found that this gene variation was there. So putting the 2 together, what does it mean? What help will this give now?
First, this is an observation. Right. But at the end, we have 2 information. First, this study points to a new pathway. We didn't know before that G protein are so directly involved in the arrhythmogenesis. That's the first part. This is a more theoretical part. But there's also a practical part. These genetic markers may help to identify patients who could benefit from ICD implantation who are not covered by the current guidelines. There are some patients who do not fulfill the criteria for ICD implantation, but they are also at risk. And these genetic markers may be helpful, but this will form a studies we have to evaluate.
Well, I thought it was interesting, though, the new ESC guidelines published this weekend in the European Heart Journal. They recommend that DNA analysis be a component of post-mortem assessment in young sudden cardiac death victims.
Cardiac death patients, yes.
Yeah, I thought that was really interesting, to help ID a genetic cause. Yeah. So, I mean, that kind of goes with what we're talking about in terms of we need to find out what those markers are.
Yes. Yes, but in young patients, in older patients, often there's an underlying disease, like coronary artery disease.
Right.
And our study was done in these cohort of patients. But in the long run, perhaps there will be a recommendation also do this genetic testing in older patients. But this will further studies have to evaluate.
So what are you doing next?
The next step we will focus on patients who are in something like a gray zone. We don't know if they profit from an ICD, but we know that they are at increased risk. And in these patients, we will apply our genetic marker and we'll evaluate if our genetic marker is helpful.
Well, we have a lot of coverage from the ESC meeting, so please check out both publications, CardioSource World News and CardioSource World News Interventions, and of course with us online where I'm the executive editor, Rick McGuire.